Great. So one last question. As you look around the world, and you've had experience in your business units in China and Brazil, clearly it's a European company, and so Europe itself is always an interesting marketplace in its own right. What do you see as the sort of timeline of development in the differences that you're seeing across the planet in the adoption of these kinds of technologies?
Well, as I said in my short introduction, the US has a head start because you already started 10 years ago with your kind of drive towards bioethanol, which in many ways had paved the way for this. But China is very interested and catching up, and we work in China with Sinopec, the oil company, and a company called COFCO, which like to be the ADM of China. So it's an agricultural commodities company, and they are both state-owned enterprises, and they have been entrusted by the government with making cellulosic bioethanol happen in China. We have together, the three of us, reached the cost targets, so we are now on par with gasoline in China for a variety of reasons, one of which is gasoline is more costly in China. And they are building their first demonstration unit as we speak. And as you know, its implementation in China can be exceedingly fast if they want to, they do it. So that's China. Brazil was actually the pioneer, of course, in bioethanol. And in Brazil, we work with the Sugarcane Growers Research Organization. They are building their first bolt-on cellulosic facility to a sugar mill. And the opportunity in Brazil is to double the output from the same cane because there's as much cellulose as there is sugar. Brazil has a very interesting vision, which is that they believe that they alone, Brazil alone, will be able to provide 10% of world's gasoline in the form of bioethanol.
As an exporter?
As an exporter.
Well, this is a good point to transition. I wish we had twice as much time as we do. But it is interesting to see how China and Brazil are leading that charge. So I will jump to the podium for something completely different. With the little time that remains, I want to talk about the reinvention of life itself, because it does sort of beg that question when we look at some of the technologies that are in their gestational phase today. I'm an electrical engineer by training, but I'm fascinated by the information systems of biology. In particular, there's so much we can learn from the code of life, how to build resilient, complex systems. And for a sense of how different this world could be in the future, compare the way we build complex systems today. If you took the human genome and burned it on a CD, it would be smaller than Microsoft Office. Right? It says a lot about both organizations and the compilers and interpreters that we run. And we have this exponentially growing capacity in our capabilities to decode, which you've heard about, but also re-engineer the code of life like never before. And I want to share some of that enthusiasm with you today. So we are entering an era where we will read and write DNA as if it were a computer program. So this will start, of course, with simple cell organisms, which you heard a little bit about today. And we'll move in applications to fuels, chemicals, food, even vaccines in the near term, and eventually waste remediation and water purification as well. So let me show you a visual. Got to push the slide. Eric Schmidt described the data explosion that we're underway. Here's a visual from the gene reading front. On the left, you see what in many ways was the exponential pace of progress that was set by the Human Genome Project. It looks like a nice smooth exponential, and people might say, well, gosh, wouldn't that cause for pause and reflection? But if you look on the curve on the right, the Human Genome Project, the left curve, fits in that little green wedge on the bottom left. In fact, it has exploded. The scientists, Venter and others, have moved on and turned their attention to the microbial world. And much of this gene sequencing, these are all the genes submitted to the public gene banks, has exploded like never before. These microbes, by the way, are abundant. Over half the biomass on Earth is microbial, whereas animals like us are less than 0.1% of biomass on Earth. And in fact, we are mainly bags of microbes. If you look at it on a cell count percentage, you're 90% microbial, mainly bacteria. If you look at it on a gene count, you're 99% bacterial. They digest your food, they cover your body. Every one of you has unique bacteria in your lungs that are evolving in real time with your immune system. It's pretty amazing. But surely this curve would have flattened by now, right? But the amazing thing is in the last year alone, one research group led by Craig Venter has grown this data set 10x again. So again, 90% of all known genes on the planet discovered in the last 12 months by shotgun sequencing entire ecosystems of organisms as he sailed his boat around the world. One of the best boondoggles ever. What has made all this growth possible? Well, It's basically the exponentially declining cost curve for these technologies. Now, don't try to read the axis. All that one can compare here are the relative slopes. Red is Moore's law. As we know it, there's a logarithmic scale, so a straight line is an exponential, and slight differences in slope are a dramatic difference. You heard earlier today that the cost of gene sequencing, meaning reading the code of life, is beating Moore's law. That's the blue line. But even more dramatic still is the green line, the cost of writing the code of life. This is dropping precipitously and has led to predictable accomplishments. The first virus created, the first living organism created that self-propagates, whose parents are a computer. This is a pretty bold new world that we're entering in. And even though this data set has exploded, as I mentioned in the prior slide, the last 90% of that data, we've lost the host organisms. We sequenced the whole ecosystem. We didn't bother to say which animal it came from. We just have the data. We don't have the organisms. Now, why would you ever collect the data without the organisms? In Biotech 1.0, you couldn't cut and paste genes from a data set. You had to have an organism to physically cut and paste from. But that's all changed, of course. Today, you can send an email file. Give me an A, give me a T, give me a G, give me a C. You send it to any of these labs scattered around the world. They'll FedEx you back those oligos you can stitch together. Now it's been demonstrated to 1 million base pair long genomes, soon about as long as you'd want. And you can boot up an organism from the code alone. That's a pretty profound change in what I call Biotech 2.0, where code comes alive. So I'll push this one more time. Consider for a moment what teenagers are doing all over the planet. There's an international contest where they get together and reprogram E. coli like it was Lego bricks. They make them into logic gates. They can make them flashing repeaters, chemical sensors, binary microbes that are used as therapeutics in Crohn's disease. They even make biofilms that literally are like photographic films. You expose them to light and they expose what I would call an E. coliroid. And these kids are doing this as if it's computer science 101, no big deal. But then consider Craig Venner's recent accomplishment announced just a couple of weeks ago, a couple of months ago on the right-hand side. They did boot up life finally from the data. It's been a 25-year quest of his, where they take 100% of the DNA out of an organism, then insert a synthetic bolus of DNA, again, created in a lab from beakers of chemicals with no animals involved, and boot up that cadaver into an entirely new species. It's a form of genetic alchemy, changing an organism as different from mouse to man, but in the microbial world. The software, in this case, makes its own hardware and reproduces and creates trillions of progeny. In the near term, of course, the complexity of systems biology means that we will continue to work on known systems from biology. We're not creating new animals from scratch, don't get me wrong. We are, in fact, primarily inventing new processes more than new products at this stage. But we can run many more experiments today. George Church at Harvard has shown that you can create 4 billion genetic variants in a single day. High throughput experimentation, if you will. And it begs the very important philosophical question of whether we will, in the future, design our complex systems or use directed evolution. Very different methods, very long conversation could ensue at the power of each method, often confused and conflated. But some companies like Genomatica have found ways to merge them. I'll give one example. They found they could make a 20,000-fold improvement in the industrial yield of a chemical of interest, much like what we heard just before, not by designing it. Human design, intelligent design got them only so far, about a 5x improvement. They instead crippled the organism, removed all redundant metabolic pathways, left only one intact, which happened to make the chemical of interest as a byproduct. It wasn't a particularly good solution. They, though, then skimmed the fastest growers over thousands of generations, just took a couple months, and they had a 20,000-fold improvement in the chemical of interest. Again, 1,000 times better than human design. They don't know how it works. They don't care how it works because the beauty is it's robust. The organism grows faster than it otherwise would, and it doesn't tend to evolve away from making the chemical of interest. So I will just conclude with a quote from Danny Hillis, who's a computer scientist, and he wrote an introductory book on computer science. And he concludes his book with the following quote. The greatest achievement of our technology may well be the creation of tools that allow us to go beyond engineering, that allow us to create more than we can understand. Thank you.